Our last session is titled How Creatures Adapt, and it's all about what's really going on when creatures' bodies adjust to changing environmental conditions. Okay? This is really what Darwin adopted back in 1800s when he published his book on the origin of species, which doesn't describe the origin of any species somehow. But he told a story. He told lots of stories. And the story that he told was that basically nature will deselect the unfit by killing them off, and then only the fittest will survive. You know, the subtitle of his book, On the Origin of Species, or The Preservation of Favored Races and the Struggle for Life. As though all life always struggles. That's a philosophy he got from Thomas Malthus. He did not get it from observing biology. Anyway, but what he said was, it's the struggle and the death, and it's the struggle and the death of the unfit dying off, and that's what makes way for new species, and eventually, given enough time, chance, and death, the new unholy trinity. Time, chance, and death. You can go from a worm to a human. And so that's what I was taught. You came from worms, which came from germs, which came from stardust. And all through this mechanism, this nature selecting by deselecting the unfit. And then in the 1930s, this ran into a huge problem. And biologists were really fighting one another and saying, okay, that's great that you can deselect the unfit, but how are you going to actually make any change without some mechanism to generate new traits? You got to have some difference, some change to take place so that you can have a difference between those that are fit and those that are unfit, so you can kill off the unfit. And they said, we don't know what difference. Darwin himself said, whatever difference it might be happening, it can't be random. But that didn't deter this group, so they in the U.S. they had a big meeting, 1930s, and they invented the neo-Darwinian synthesis. And with the NDT synthesis, they said, it's mutations, which had been described by Hugo de Vries, the word invented in like 1901. He worked with the evening primrose for a decade and a half, and he found, there's a trait that just suddenly pops into existence, and it becomes part of a true breeding line of these plants. I'm going to call that a mutation. And so these guys just decided this is how evolution is going to have to work. The mutation emerges, and then that gets selected by deselecting those who don't have the mutation. So now mutation becomes like the new savior. Okay, so now ever since then, biologists have been looking for what is the mutation that caused this creature to have a change and a shift so that it can be the one to survive and thrive and push evolution forward through time. And they keep, guess what? Not finding the mutation. And they keep not finding natural processes doing any selecting. And yet they continuously frame all these changes in terms of neo-Darwinian theory, even though the facts don't fit the theory. So we're going to look at some of these examples. Here is our ex-biologist Scott at the Institute when he did his mosquito project. On the one hand, we're not happy that he stopped because he stopped before he could publish the results. But on the other hand, we're thrilled that we no longer have leaking mosquitoes that fly into my office. Because he was in building B with me. We have 4 buildings in our complex at the Institute. Anyway, he had the mosquito lab. I would just say, Scott, how do you do that? And he's from Fort Worth. Oh, after a while, you just get used to it. They's hungry little buggers anyways, I could not do that. And that's why we haven't finished these results. So everything he did was preliminary, proof of concept, but it was stunning what he found, which was basically that mosquitoes, he first did his literature research, which is how you start any science project, and he discovered that there's over 100 species of mosquitoes, and only a handful of them actually take blood meals. The rest of mosquitoes all take meals from flowers and fruit. And it's only this handful that takes blood meals. And then he started researching how do they know where the blood is, because it's behind your skin. And it turns out that that not only do humans emit CO2 and that these critters have CO2 detectors, but so do flowers and fruits. Not only is our temperature higher, and they have thermometers, and they can detect temperature differences, and that's why they go to your body as well. Flowers have temperature difference. The flower actually emanates a little bit more heat than the surroundings. So all the features that these critters can detect, which my evolutionary colleagues have for many years insisted, had evolved in this critter to enable it to take blood meals. Evolution has produced its blood-sucking capabilities. Turns out, nope, those capabilities have always been used since the beginning for very good purposes and not painful ones. My wife asks me, periodically, every time she gets a mosquito bite, why did God make mosquitoes? And I understand that question. And early in our marriage, I would try to explain, especially after this project got going. I was like, oh, sweetie, there's a new answer. Why did God make mosquitoes? It turns out and I'll explain it. Most mosquitoes, they just take plants, and so God originally made all the mosquitoes very good, part of the very good creation, Genesis 1:28. But now, because of sin, now some of them take a blood meal, even though they weren't originally meant to do that. And then you know what her answer is? Why did God make mosquitoes? She does not want my answer. All she really wants is for me to say, I'm sorry, dear, did that mosquito bite you? So now I have learned to show sympathy instead of supply science or whatever. All that to say, you'd think that, we ordered these mosquito eggs from a lab, an evolutionary lab that had been breeding mosquitoes for 30 years, many, many generations of graduate students sticking their arm in the net and giving these blood only for 30 years, maybe hundreds of generations. You'd think by now that the natural or the unnatural selection of blood only would have made these critters evolve into blood only, specifications in their body structure. But no, when Scott found out that most mosquitoes like to have nectar, he made a nectar concoction, placed it in the mosquito netting during feeding time, stuck his arm in there at the same time. All 100 mosquitoes went to the nectar. Not one of them took a blood meal. After all those years, they still preferred their original diet. But they had been adapting, using their God-given abilities to take whatever they could to survive and make the next generation. And so what we see here are examples of creatures not showing what evolution expects us to see. And the mosquitoes tipped us off that there may be something else going on in these creatures. So our president has come up with a new theory of biological design set to replace Neo-Darwinian theory. Neo-Darwinian theory has one central tenet at its core, other than the fact that there can't be a God involved, and that is that it has to be the universe, the external, the outside. It's the environmental factors that are shaping and modeling organisms as though the organism itself is passive, modeling clay, waiting for sunlight, waiting for predators, waiting for climate to make changes to it as it moves through space and time over many generations to convert that worm from a germ and into a German, if you will. External. And what we're saying is maybe we should look internally. What's going on with the internal mechanism? What features do these creatures use when they actually adapt to changing environments? Do they have detectors? If you were going to make a drone that flew on autopilot, what kind of features would you want to put into your drone? You know, these things that fly and they're annoying sounding, like a giant mosquito or something. You would want to have it be able to see its surroundings. So you'd want to install what? Cameras. Yeah, cameras. You'd want it to see when the lighting is bad, so you'd want to give it radar. And then if you put cameras on it and radar on it and didn't tell the machine what to do in response to the input, what good would the cameras do? Nothing. So you have to have a processor in there to tell this drone, okay, if you see mountain ahead, then do X. Increase, the fan speed so you can go up and over the mountain. Or some kind of, reasonable response to this input. So you need detectors, logic mechanisms, and call those processors, and then appropriate responses. Are critters doing this? So that one generation can detect this and respond in this way, or tell the next generation through internal mechanisms, hey, kids, you're going to encounter what I just encountered here, so you better develop into a body that's going to be able to process that and live life a different way. And then it should be repeatable. In other words, if this drone comes up to another mountain, it should be able to do the thing again and make these changes again. And what we're finding is that critters actually do this kind of thing, and they do it across generations, and they do it repeatably and reversibly. So it changes, and a few generations later, it might change right back to the status of the original parentage of that lineage. Okay, so we got this many examples. Count them. And the first is the peppered moth. Once an icon of evolution, still in the textbooks, I bet. Biston betularia. What a cute name for a cute critter. Anyway, there's all kinds of different species of peppered moths. This one is in the UK, and the story goes, in the mid-1900s, some scientists and biologists were examining these moths, and they looked at the moths before, they thought about the moths before the Industrial Revolution, which in the UK was pretty intense in the late 1700s with all those mills and stuff. So before the Industrial Revolution in the UK, we had lots of white-trunked trees. I don't know the names. Birch, I don't know, something. And then beech, I don't know. Then the moths would land on the tree trunk, and it was mostly white with like pepper sprinkles of dark on its wings. And so the birds couldn't see them. It was good camouflage, and they didn't get eaten. But after the Industrial Revolution, soot was in the air. The soot stuck to the tree, made the tree trunk from white to dark, and now the white moth lands on the dark trunk, and the bird can see it, and the bird does what? Picks it off. Now we have the death of the unfit, and now we have a model and an example of evolution. So that today, there's many more dark-winged peppered moths than there are light or whitish-winged peppered moths. That's the theory. But there is a question about that in terms of it being an example of evolution. Yeah, the critter changed, but did it change because of the death of the unfit? Did it change because of predators? Really, was it an outside influence? Is that all we are allowed to think about? After all, before the Industrial Revolution, what did we have? We had some white and some dark. After the Industrial Revolution, what do we still have? Some white and some dark. Just because you shifted more of them dark and more of them light doesn't mean you've actually done any real new information. We haven't actually done any unidirectional, helpful evolution. But now the geneticists get involved. Oh, those silly geneticists looking at the insides. What are they looking for when they look at the inside? What mutations have happened to turn these moths darker? That's the question. And what does the question presume? It presumes that these critters evolve by mutation and selection, period. That's all they have in mind. Guess what they did not find? They didn't find the genetic difference. They didn't find mutational differences, but instead they found a different kind of genetic shift, and it's called transposable elements. So it's just a cut and paste. You guys have cut and paste words in a Word document from one sentence to another. It turns out that these moths have an inbuilt ability to cut DNA sequences and paste them into very specific places in their genome. And they tested all kinds of moths, several hundred moths this way, and they found that 103 out of 105 dark moths had the same exact transposable element copied and pasted into the same exact spot over and over again. And what that did is it lengthened, it changed the shape of the gene or the information on the gene so that during development from an egg, these critters, or during development, these critters were directed. The coding instructions to make pigment, put the pigment into the wings, was changed. What did the change? Well, they said this in their paper published in 2019 in Biology Letters. Our results imply the conservation of a developmental master switch for melanism, which is the pigment, melanin, in geometrid moths, which includes peppered moths. They did not say our results imply that predators changed the development in geometric moths. No predators. That was all a tale. It was all just a story about birds and death. What's really going on is these critters are detecting something in their environment, and they're telling the next generation of moths somehow, you better make your wings darker. And so then the next generation of moths deploys this genetic altering mechanism to shift the code so that the code goes from lighter winged to darker winged. And it's all by internal, not external mechanisms. Wow. So there we have the first discovery that fits this model of internal adaptation through design. Not adaptation by accident. They were designed to adapt, is what we're thinking. Well, let's look at the next one. Genus Ariamnes, the stick spider, the Hawaiian stick spider. And here they are on the islands of Hawaii. We have on Kauai, so Kauai, Oahu, Maui, and then Hawaii from top to bottom. And these are arranged from west to east. And that's the order that the islands actually formed. Relatively recently, by the way. And you got a red one on one island, a white one on the next island, a gold one on the next island, and a black one on the big island, brah. So my daughter married a man from Maui. They moved to Maui, so we had to go visit our kids in Maui. Whatever. And so I went looking for the stick spider, and I looked and looked for like hours. I'm like, this critter it hides really well. I couldn't find it. I went back home in Dallas and read more about it. It's nocturnal. Well, I tried, and it was fun to try. Now, how did these critters do this? You'd think that if evolution were the cause, through mutations, natural selection, that you'd have to have one of these spiders evolve the black version, and another evolve down the lineage a white version in the other colors, and then the black one would have to hop to the next island. The white one would hop to the next island separately, and then as they're hopping islands over the eons that supposedly elapsed since these islands formed, you would have accumulated mutations in these different colors and these different lineages. But these biologists publishing in current biology found something completely different in the genes and in the genetic structure of these spiders. What they found was, check this out. So here we have across the top all the spiders in Kauai, red, dark, and gold. Here we have Oahu, Oahu, Oahu. That's where Honolulu is. White, red, and gold. And then here, on Maui, gold, red, and white, red, and gold again and again. And it turns out that, look, these different ones here, these different looking ones on Maui are genetically identical. They're genetically the same. And so what happened, and these are more similar to each other, even though they're different colored, than they are to the ones of the same color on the different islands. This is stunning. This is amazing. So what must have happened is that one spider pioneered the next island over, and then it diversified into its pre-arranged suite of options. It had kids that were gold and black and white, just maybe like that. And then another spider did the same thing on the next island. Another spider, same thing on the next island. So that all the spiders on each island are more genetically similar to each other than they are to the other spiders, or they're the same color on the other islands. So they're not diversifying, they're not deploying these very different looks because of any kind of nature selecting mutations or anything like that. It's just because they were designed to deploy these different options available to them very quickly. It's like a Swiss Army knife, genetically speaking. This generation needs to be red. Well, we need the can opener. Next generation, we don't need the can opener anymore. Let's fold that down. We need the screwdriver. So you have this genetic toolkit where you have the The information for the gold version is bound up in the black one, or vice versa. But it's only deployed if and when the triggers say, deploy this version, deploy that version. So it's these different body options in these little critters. And you can enjoy me talking about this even more on YouTube, where we have a series called Creation on Location. And there I am appearing through the jungle on Maui, looking for, but not finding these spiders. I didn't find them, but I was able to talk about them and how much they confirm this quick and repeatable adaptation. Each island, they repeat this redeployment of the same suite of options. The white one lives on lichen. You know what lichen is? Lives on tree trunks and such. The black ones live on the lava rock. The red ones live in leaf litter. So they each know where to live. They each have their own niche within one forest, even right there on those islands. The islands, brah. Lots to learn. Now, the next animal is, do you like animals? Oh, you're in the wrong place if you don't like animals. Today's Animal Day. It's Animal Afternoon on a fine Sunday. This next animal is the blind cave fish. In particular, it's the Mexican tetra. So here we have the Mexican tetra Astyanax mexicanus. And we're doing all kinds of experiments with these at the Institute. You can read about it through subscribing to our magazine, Acts and Facts, or just go to our website, icr.org, and type in Mexican blind tetra. And what we're finding is that these critters, although they look very different from their sighted eyeball-having counterparts, they're genetically the same. And they interbreed. When these were first discovered, back in the 1920s, I believe, they gave the sighted ones with the eyeballs a particular name, and then they gave the blind ones, which have no eyes, very little pigment, different feeding strategy, different physiology. The blind ones have larger red blood cells, and they have a larger gill-to-body ratio, all kinds of differences. They even have the blind ones have enhanced lateral lines, which is their sensory mechanism, which goes from here down the sides, and they sense pressure. And we can watch these fish swim in our fish tanks. You don't have to go to Dallas to see this. Just order some fish on Amazon. They'll send it to you. And a couple dollars per fish, and then put them in your fish tank. They avoid each other. They don't even need eyes because they're sensing the pressure differences that are around them, and they interpret those pressure differences to generate some sort of an internal map where they know exactly where to go, what to avoid. And it's fun to watch them swim without eyes. And they have enhanced chemical sensors. So that's a whole suite of very helpful traits for living in a cave. How did they get there? When they first discovered these, they gave them two different scientific names. But then some researchers thought, oh, well, we'll just have them swim around together. And suddenly they interbred. And then the first generation had pigment and little tiny eyes. And so they still interbreed to this day. So we know that they are descendants from the same basic created kind. In fact, there's hundreds of different species of tetra, and probably all of them belong to the same created kind originally. Well, they would have survived the flood outside the ark in those floodwaters and made their way onto land in freshwater, and then subsequently made their way from surface freshwater into caves where there's very little food and very very few ions, which means low conductivity, which means that affects their physiology in different ways. That's what we've found out. Okay, so what did we do? We've done scientists publishing in 2013 in the journal Science found that when they took the eggs of surface fish and put those eggs in a simulated tank, a tank that simulates cave conditions, what they did is they lowered the ion concentration, and it turns out that they were able to measure on the eggs, the egg shell, which is transparent. But it's loaded with sensors because the eggshell was detecting the electrical conductivity of the water around them. And then that sent a signal to a signaling protein called heat shock protein 90 or HSP 90. And that sent a signal down into the nucleus of the cells that are developing into this fish from an embryo. And that signal said, warning, you're going to have low conductivity. And so the resulting fish from that experiment ended up with really, really tiny eyes. So all we can imagine is them doing that a few more times, and their eyes would get smaller, because they have that option available to them, and it's not even genetic different. There's no mutational differences between these fish. They're just adapting, and they're deploying the different adaptations. The experiment that we published 2 summers ago was where we took the blind cave fish here, a lovely couple, guy and a gal, and we had them in a tank that simulated surface sunlight. Now, Mexican sun is hot, and so we put this lamp really close to the surface of the tank, and at that distance, UV light penetrates right into the skin. And we thought we'll raise a generation of these blind cave fish, and we'll see how many generations it takes for, if they still have the genetic capability or the instructional coding to make pigments, maybe the pigments will come back into play in some future generation. 30 days later, those very fish, having descended from who knows how many countless generations of pink only fish, suddenly were just as tan, just as pigmented as their surface fish counterparts. Don't have pictures. I should have them. I need to get them. But we have those. So we have those fish swimming around, and they're blind cave fish, but they're not pink anymore, because suddenly it's like they got a suntan, and they're ready to deploy that pigment to protect their DNA from the ultraviolet. It was all sitting in there. It had not been deselected by any kind of natural selection process. Anyway, these critters deploy the cave option or the surface option, not because of DNA differences, but because of something called, in large measure, something else called epigenetics. Have you heard the term? Couple people? Epigenetics? Think of the DNA information like a set of encyclopedias. And let's say, what do you use an encyclopedia for? Well, I want to learn about this. I want to learn how this thing works. So you go to the encyclopedia and you go to that thing that you need to learn how it works. Well, if you don't need to know how this thing works, you don't ever want to know how this thing works, then what do? A good mechanism would be to take a big paper clip that section off so you don't have to worry about it. You're never going to need to access it, at least not in your generation. So the epigenetic is a coding system that puts paper clips on the DNA to mark off these sections of DNA, telling the resulting cells that develop into the adult, you don't need this information, you don't need this information, until you do need the information. I mean, if you tear it out of the book, then it's gone. And subsequent generations, if you copy your encyclopedia and hand it to the next generation, they don't have that information anymore. So much better to just put a paperclip on it in case some future generation might need that information. That's what epigenetics does. And it's an ingenious layer of information that closes off and opens up access to and from DNA that's in these. So anyway, we know these critters do epigenetic coding so that they can deploy these trait differences. Okay, let's look at the three-spine stickleback. By the way, what did we learn from that last one? These eggshells have detectors, and the detectors are detecting the conductivity of the water, and that's critical to the formation of the right kind of fish. Three-spine stickleback is another example of, supposed to be an icon of evolution, and in this critter, they have a freshwater version, so three-spine stickleback. And it's named gasterosteus because near its stomach, or its hips, it's got these bones. Osteon means bone, so it's got these hip spikes like this. I'm glad I don't have them. This guy does. But it's the marine variety that has the big hip spikes, and the freshwater variety, the hip spikes are diminished or gone. The marine variety has three big bony plates, one on each side. The freshwater variety has only two or maybe one, depending on which one. Now this map shows the number of places around the globe where this fish has pioneered from the ocean, marine environment, swimming up the stream, finding a pond that's now freshwater, and pioneering that freshwater pond. They've done it in Scotland, they've done it in Alaska, they've done it in all these different places. near Alaska, sorry. Is it in Alaska? Yeah. And so what's been going on with these fish? Have these fish been swimming up, and then why do they turn into the freshwater variety? Where do the spines go? Why do the spines go? What's happening? Is it because of the death of the unfit? Like, what's happening here? And then the geneticists got involved, and they start looking for the DNA mutations that are supposed to be causing this. They didn't find any mutations. What they found is reuse of a gene. Reuse of a gene. Folding the gene away, putting a paperclip on it, and then later generations, let's reuse that gene. Taking the paperclip off for the kids so that they can thrive in this new freshwater environment. Look at this quote. Reuse of the EDA gene provided the initial evidence that the repeated evolution, I don't think it's evolution, I think it's just repeated deployment of a designed mechanism to adapt. I know it's a lot more words, but it's at least more accurate to what's going on. Of freshwater traits was the result of the death of the unfit. Oh, wait, sorry, no. It was the result of natural processes molding and crafting these critters. No, it was the result of 3 key words here: standing genetic variation. The genetic variation was already standing in the creature. Well, who put it in there and when? I have an idea about that. It's called the Creator, the Lord Jesus Christ, who made these creatures with standing genetic variation so that they had the capability to adapt to either marine or freshwater environments. Boom, right there. It's the redeployment, reuse of this gene. That's what they found. That's what these evolutionists found. And it's really consistent with our creation-based model that creatures were crafted to adapt. Here's another study on the same kind of fish. And there they are. There's an x-ray of them. Which one is marine, would you say? The one on the bottom or the one on the top? Yeah, if you say top, you get an A for the day. So there's the marine one. It's got those big spines on its pelvis, gasterosteus. Anyway, these guys showed, so what they did is, before you read the quote, just listen. They were tracking this population of sticklebacks. They had a population of sticklebacks where there were no predators in the pond. Then they introduced predators, other fish that like to eat sticklebacks despite the spines. Well, hey, they're freshwater varieties, so they're not very spiny anyways. And this is what happened. So they analyzed the genetic, not just the genes, but how the cells are accessing those genes, when the cells are accessing those genes, and they actually now have ways to measure how often the cell is accessing certain genetic regions. It's just remarkable what biologists can do now. Anyway, through all that, they showed that repeated exposure to cues of a predator, and I'll explain what that is, results in significant differential expression of genes. So now they express a different suite of genes at a different rate for different tissues, and these genes are involved in immune response, synaptic processes, brain metabolic processes, and visual perception. Look, if Uncle Bob is getting eaten, if your neighbors are getting eaten by this new predator, wouldn't it behoove you to know that there's a killer on the loose? Wouldn't it be great to know something about, and then to do something with that information? Hey, there's a killer on the loose. We better keep our eyes open. And certainly that's exactly what happens within hours. These creatures have enhanced visual perception. And so the eyeballs are looking up, they're detecting more information from above them where the predators might be, and they're processing that information faster, and their neurons are firing faster, and they're putting more resources into the eyes and into the brains than into reproduction or whatever else they might be doing, because they want to know that there's a killer on the loose. How do they know? What is a predator cue or an alarm pheromone? Other writers put it that way, alarm pheromone. Well, if you're a 3-spine stickleback and Uncle Bob, your 3-spine stickleback uncle, gets eaten by the predator, it's not that long after that the predator poops out bits of Uncle Bob. And those bits of pooped out Uncle Bob that are in the water floating around, they become the alarm pheromones. Now, are the alarm pheromones, when they drift down into your chemical sensors and you go, that's a bit of Uncle Bob? does that mean that those alarm pheromones are actually causing your differential expression of genes so that you have enhanced synaptic processes? Not at all. Those are just bits of Uncle Bob. They can't transform anything. But you already have the mechanism built in to detect bits of Uncle Bob and to do something about it, which is to look out, there's a killer on the loose, and he got Uncle Bob, and I don't want him to get me. Internal, repeatable, specific responses targeted to the need at hand, as though it was designed and divinely and put in there in place on purpose from the start. Wow. Just 3 more, guys, and then we'll do our last Q&A. And I am really enjoying today. I hope are you learning stuff today? Oh, good. Excellent. The ground finch. This is famous Darwin's finches, an icon of evolution. What's going on with these finches, and why do they supposedly support evolution? By the way, lately we have to call them tanagers because they were originally miscategorized. So Darwin's finches are now tanagers, but we're still going to call them Geospiza for their genus name. And this is the next story to be told. There we have a version with a long beak on one island. These are near the Galapagos Islands. These are the islands. Yeah, these are the Galapagos Islands. And you've got a version with a short beak and a version with an intermediary beak. And it turns out that over the generations, next generation can deploy a shorter beak, and then generations after that can go back to the longer beak. So here we have another example of no net evolution taking place. Even if you want to call these subtle shifts in beak shape, if you want to call them evolution, you're not really going in a direction. What I need to see, if evolution is true, I need to see a creature that has no beak evolve a beak. No wings, evolve a wing. No feathers, where's the feathers? We don't see any of those kinds of transitions. All we instead see are these subtle but very helpful transitions, and they don't transition so short that the beak is useless. They don't get so long that the beak is useless. It stays within a useful range. We see this over and over with all these different kinds of creatures. But is it really that the critters, the birds with the long beak in seasons of drought, their beak is so long that it can't crack the nuts, so they die, and only the birds with the short beak, which now have the leverage to crack the nuts and get the food during drought, and that's how they survive? Is it really the survival of the fittest going on here? It turns out that some scientists, these geneticists, these developmental biologists, they get involved and they ruin the evolutionary story. They found this. Eureka. Isn't this fantastic? You came here to get blessed with a whole bunch of squiggles. I know you did. I'm told that this formula describes the shape of 2 semicones, 2 half cones. So if you're going to make a shape of a cone, you can either draw it or you can make a formula, and each of these letters is a variable that represents a suite of numbers. So you got 1 half cone here and another half cone here, and if you bring them together, you have a whole cone, but with 2 half cones, you can also open them and make a beak. Okay? So that's the math formula. They got this from the geometry people, okay? And then they went into the biology, and they used this special technique that we now have at the Institute with our laser scanning confocal microscope to look at, you can see, can you see? These are little, this is inside the egg, inside the little developing embryo, and each of these little different cell types is colored a different color. And it turns out that the green cell types that are wrapped inside this red shape, they're building a new beak out of nothing, or out of yolk. They're building a beak out of yolk. And so it goes from early development, middle development, later development, and they're tracking the distances, mapping out, like, how do these cells know where to put new beak? Why don't they put the beak on the butt? Like, they know where to put the beak, and they know exactly what shape the beak's supposed to be. And what they found on these graphs that show is that the cells that are making new beak, they make the beak according to this exact formula. So the cells have this big fat math formula baked into them genetically. And all it takes is the adjustment of one of those variables in order to get the shape of the cone either a little bit longer or a little bit shorter. and that's it. And the adjustment of that variable involves no genetic changes. It's the same genes for the beaks, birds with this beak as the birds with the long beak, same basic genes. And so it's not a genetic difference. It's, again, some other coded information that tells the cells which genes to open up and when and how long and how often and in which tissue and in what time of life, early in life or later in life. And all that information is already baked in there. Oh, wow. No death of the unfit, no mutations. It's just a story. These critters are changing their beaks on purpose, pre-built. Deer mouse, and it's, again, internal. We got to stop thinking that the world can do this stuff, because really what that is, it boils down to the same thing that Paul said in Romans chapter 1. We worship and serve the creature rather than the creator who's blessed forever. Amen. And we as a church have bought into this way of thinking that worships and serves the creature or the created rather than the creator, giving the created inanimate factors of the world, these environmental processes, the power to generate new creature traits and whole new creatures. Wow. And we buy into this stuff, and I did for many, many years, until I started studying what I get to study at the Institute and realizing and seeing, these creatures are actually doing this because of internal designed, well-designed capabilities. And then my worship of Jesus, you know what happened? Jesus was kind of like really great, and now he's greater. And I worship my worship of him has elevated because of my study of biology, because he gets credit not only for crafting the basic structure of a bird, but for crafting that bird to be able to, first of all, copy itself, wow, make a new generation, but then to tell that next generation, hey, you might want to have a longer beak or a shorter, to have variations that are appropriate and helpful. And that's how creatures are adapting in many cases. For example, the deer mouse, Peromyscus maniculatus, more deer mice in the world than humans. Or maybe it's North America. The history of deer mice is that they started in the center of the continent, and then some went east in one lineage, and another lineage went west. And the ones in the progenitors, or the parents, had a tail of a certain length, but when they pioneered the forests of the west, the prairies of the west have short tails, but when they got into the forests, suddenly the population that now inhabits the forest have longer tails. That's pretty cool. Is it accidental? Is that because of some sort of predator that's in the forest that eats the short-tailed ones more often, and so you have the death of the unfit? Is that what's going on? Probably not. Because here's what's happening out east. Same exact thing. Different species names, but it's the same critter. It's just deploying a trait difference. Now, these tails are longer. Why? Because in both cases, whether it's the eastern forests or the mice that inhabit the western forests, each vertebra is a little bit longer, and there's a few more vertebrae to make the tail longer. Possibly it has to do with helping these critters balance on branches because they're now in a forest. We don't know why they have longer tails. Maybe there's a functional reason why, but they're redeploying the same trait to inhabit and to pioneer these new places. And why not? After all, what did God say in in Genesis chapter 1 to these critters? He commanded them to be fruitful, multiply, and fill. Fill. Go fill the forests. Go fill the prairies. And wouldn't he, if he's kind of nice, wouldn't he equip these critters to be able to fill, be able to pioneer? Go fill the cave waters. And I'm going to give you a suite of body options so that you can live and even thrive in caves. And he did that for all these cave-dwelling critters, not just the fish. Yes, he provided all this. He even provided the ability to use this Swiss Army knife. Long tail version, got it. Oh, short tail version, fold this one back. Swiss Army knife, fold it out. All by design and all to the credit of the Lord Jesus Christ, our Creator. Well, lest ye think that I am saying or that I believe that all creature changes, all these adaptive changes are through this process of detecting and deploying. Not so. Perhaps most creature changes are ornamental, not fundamental. Maybe they're ornament. In other words, yeah, it's helpful for the critter to thrive in this environment, but you know what? Some birds are just pretty. Some flowers they look pretty. And they have different colors. And I think God looks at them and goes, oh, I think I need you to be purple this generation. I think it'd be cool if you could go from gray to like turquoise. So bird plumage can do that. Animal fur can do that. We have curly fur and straight fur and different looks, different attributes. And some of it is not functional. Some of it may be fashionable in God's eyes. And we have noticed for many years now that the mechanism to do that is something called alleles. Heard of them? Alleles, 3 people. So an allele is just a different version of a gene, So you may have a gene that, let's do an oversimplified illustration. Let's say you have a gene for hair color, and your gene, there's a blonde version and a brunette version of the gene. If you inherit a brunette and a blonde, one from mom, one from dad, it's not how it actually works, but I'm just illustrating. Then your cells have to decide which one am I going to use. Let's say your cell decides to use the brunette one. So you have brunette hair, but you're carrying mom's blonde version, mom's blonde allele, okay? And then so that way you can recombine, reshuffle the deck every generation, and redeploy a new suite of alleles. And it turns out that creatures inherit this suite of alleles that they share within their population, and they become stably breeding within that suite of alleles, and we call that a species, but all it is a variation within a created kind. And that's what we see with bovids. What are bovids? Okay, let's move right into this. There's longhorn. Man, them horns is long. But cows, whether the horns are long or short, can interbreed with bison, the American bison. And the offspring is commonly called a beefalo. There's beefalo ranches here and there. So they're definitely part of the same created kind. But many varieties within a kind, or subspecies or species even within a kind, they can no longer interbreed. And it turns out that God actually placed into creatures barriers to interbreeding, and basically mechanisms to encourage them to speciate or to diversify. Anyway, so that has happened with the American bison with its suite of characteristics, but it can still interbreed with modern cows. Experiments have been done between the bantang can interbreed with bison as well. The yak is basically just a furry cow that's in Tibet, and it can interbreed as well with cows and bison. But the water buffalo and the Cape buffalo, we've gotten, researchers have been able to get, I think, with IVF, they were able to get, kids, that's complicated, ask your parents. But they're able to get the cell, the developing embryo in the mom's womb, to go to like the 84 or the 168 cell stage before it aborts naturally. So anyway, there's suggestions that it may still retain the ability to come together. In other words, it may still be part of the same created kind. Maybe God created one suite of cow kinds. It would have been a clean animal on board the ark, so you would have had 7 pairs. That's our interpretation of the Hebrew there, 7 pairs. So 7 males, 7 females of the cow kind, which who knows what they would have looked like on the ark. Maybe something like that bantang. And from the bantang-looking thing, we have now bisons and yaks and maybe even buffaloes, African buffaloes as well, possibly. So, there's still work to be done to find out which critters now belong to the same created kind, but we definitely see evidence of reshuffling, redeployment of alleles, and that's how we generate all kinds of different trait differences. Question, is reshuffling and redeployment of alleles, is that an internal or an externally driven process? Internal. Who put all the alleles there? Praise God for alleles. If it wasn't for alleles, all the guys in this room would look like clones. And like we'd all look like Harry maybe. That would be a tragedy. Scary. Scary Harry. We'd all be the same. So he gave us alleles so that we have trait differences that make us unique. Each one of us made in his image. We're all human. We all have the basic fundamental traits, but we all also have unique fashionable traits that make us exactly who we are, exactly who God wanted us to be. And guess what he does? He loves every single one of us just the way we are. We have no time left. We've had fun. We've learned a lot of stuff. We've seen all kinds of evidence of internal capacity to adapt, and he gets the credit. All things were made through him and for him. You're a thing. I'm a thing. I'm a created. You're a created. We were created for him. Are you living for him? Is there anything keeping you from saying, Lord, I'm yours? That would be my challenge after all that we've talked about all day. He made you on purpose with all the specializations that you have, the tools and skills you have. And he wants to know you, and he wants you to worship him. But he doesn't like half measures. He doesn't want you to go, I'll worship you on Sunday, but on Monday I'm going to go live life. No. Give him your whole life. You're built for him. You might as well live for him as well. You can do that just to find me at the end and say, I want to commit my life to the Lord. And I want to live for him and stop fooling around with the world. You can do that. It takes mentorship. Find another Christian. If your parents love the Lord, go to your parents and say, I want to love the Lord and worship him with all my heart for now and forever. If you still have doubts about creation and the flood, that's why we have our ministry, the Institute for Creation Research, and all the resources that we offer, including my new book, Living in Light of Genesis. It's the second best book ever written. Bible, Living in Light of Genesis. My third book, Parks Across America. That's awesome if you like pictures. And of course, we have Acts and Facts. All these things are available electronically as well at icr.org. Hey, it's been a great afternoon. I'll see if I can answer just a few questions, but I really appreciate you hanging out all day with me. Thank you very much.